Hi everyone. In this lecture, we're going to be covering Chapter 3 in the Microbiology for Surgical Technologists book. And this lecture is going to go over different types of microscopes, culture media, gram staining, and laboratory studies. Now, as surgical techs, we're not going to spend too much time looking at microbes under a microscope, but we do handle and care for specimens in surgery. So this is the WIFM for us. What's in it for me? What's in it for the surg techs, right? We're going to be helping to prepare and care for specimens like bacterial cultures, frozen sections, amputated limbs maybe, foreign bodies, tumors, all kinds of interesting things. And from a microbiology perspective, one of the things that they might do in surgery is a culture and sensitivity test. And this is where they're going to swab some strange looking fluid, send it to the lab to see if maybe there's an infection of some sort. And the lab is going to do a culture and sensitivity test to see which antibiotics that specific microbe might be sensitive to. So this is going to help them to be able to diagnose and treat the patient better. So we deal with specimens. That's how this is all related to us and what we do. Now, as far as those individuals that work in the lab, we're talking about the laboratory where they process various types of specimens, whether they're tissue specimens or cultures or whatever the case may be. The physicians and the scientists are only one component, right? But just as in the operating room, there are techs that are an integral part of the process, and it is the same in the lab. Now, for us in the operating room, we're going to be, handling the specimens, and we need to know what the specimen is called, and we need to know how to send it or how to tell the nurse how the doctor wants it to be sent. So those are some of the things that we're going to be doing in the operating room. And then that specimen is going to get taken to the lab. Now, sometimes we want to have the results right away. And so they might take some tissue to the lab and they're going to do something called a frozen section, which means we want to make sure that we got out all the cancerous cells, let's say, if we're doing maybe a breast biopsy for a malignancy. And so we're going to be waiting on that pathologist. Sometimes the pathologist will actually come into the operating room and kind of take a peek so that the surgeon can kind of let them see the specimen in situ. So we became a bit more familiar with the metric system in math, and this is the typical system that they're going to use in the lab. And where we were looking at meters and decimeters and centimeters, microbes are super, super small, so they're going to be measuring those in even smaller metric units like micrometers, nanometers, and angstroms. You can see a picture here of that scale of measurement. So now we're going to talk about different types of microscopes. So we're going to look at the compound light microscope, the dark field microscope, phase contrast and fluorescence microscopes, and then the electron microscope. Okay, so we're going to begin our discussion about microscopes with the oldest, most antiquated type of microscope out there, which is the compound light microscope. So if you've taken a biology class in the past or maybe in high school and you look through a microscope, this is probably the type that you used. And so this microscope has a 2-lens system, and it also has a light source that shines up through a window in that flat platform that you see there in the picture. That's called the stage. And then the light shines up into the ocular lens, which is where you put your little eyeball to look in there to see what's going on. Now this magnification maxes out at about 2,000 times. So if we're going to see really little critters, then we're going to have to use a different kind of microscope. However, the 40 times lens is called the high dry lens, and it's used to see larger microbes like algae and protozoa. And then if we want to get a little bit better magnification, then we can move to something called an oil immersion objective. And with this, we have to put the organism that we're looking at in a little drop of oil so that it can be seen clearer. With this microscope, the light has to be focused, and in order to focus it, we use something called the condenser, and you can see it is on this slide right here. Hold on, let me highlight that for you. Condenser is right there. Underneath there is a little knob, and you can kind of dial it in and focus it down. And one thing that kind of limits us here is the wavelength of the light. And that is really what limits us because it's a long wavelength. It's not going to have as high of a magnification as other microscopes would. Something that can help us see microorganisms at that maximum magnification is staining the microorganism, and we're going to talk about gram stains, but staining that organism changes the color of it from the background of the slide, and it causes light to bounce off from it differently and allows us to see it a little bit better. You know, that increases the contrast between the two and helps us to be able to see the organism that we're looking at. So that in a nutshell is the compound light microscope. And now we're on to the dark field microscope that really just portrays itself as a different kind of microscope, but it's really not. It's really just a compound light microscope in disguise. And the way that it is in disguise is that there's a special little opaque disc that they put over the condenser that shines the light to darken and dampen the light. So we use this when microbes aren't visible with the use of the light microscope, or it can't be stained by standard methods, or if staining distorts the microbe in some way and we really can't get a good look at it, then this dark field microscope can be an option. All right, so now we're going to talk about the phase contrast microscope. And the phase contrast microscope is really awesome because it allows us to look at microbes while they're still alive. And so we don't have to stain them. The staining process does fix or kill the microbe. So here we can look at it in real time. And it's alive while we're doing it. Something else that's really awesome about it is that it allows us to look at those internal structures in the living microbes. And because it bends light in different directions, there's two different types of rings that focus and either refract the light or undiffract the light, and because of that, the microorganism is going to appear in various shades of light and dark, and you can see that in this bottom left image that I have here for you, where the internal structures, the edges of the microorganism and the nucleus inside are kind of outlined in brighter white light with varying shades of gray in the background. So this allows us to see the internal structures of those microorganisms. It's pretty awesome. Here we are looking at the fluorescence microscope, and the fluorescence microscope allows us to look at microbes that are either naturally fluorescent or we can stain them with a group of dyes called fluorochromes. And then these microbes are going to appear in these like really bright psychedelic colors against this dark background. And one of the primary diagnostic techniques that they use with the fluorescence microscope is called immunofluorescence. And this is a process of injecting some sort of specific antigen, like a bacteria, into an animal, and that animal's immune system is going to produce an antibody. Remember, we've talked about this before, and those antibodies are going to be a specific lock and key, right, to fight against that antigen, fight it off. So they're going to extract some of those antibodies from the animal, and they're going to add fluorochrome to those, and so now they're going to be fluorescent. They're going to put them in the scope, and they're going to add some unknown bacteria, and if it's the same bacteria that was in the animal, then they're going to produce that same exact type of antigen, and that antibody is then going to attach to that antigen, and they'll be able to discern if it's the same type of bacteria or not. Certain fluorochromes also have an attraction to specific microbes. So here on the screen that you're looking at on the far left, that is anthrax. Anthrax stains a red-green when they add fluorochrome, and then the bottom right little image is tuberculosis. It kind of stains a bright glowing yellowy green, and there's two main types of immunofluorescent dyes, fluorescein, which stains yellow-green, and then rhodamine, which stains red-orange. So there are two types of electron microscopes. There is the transmission electron microscope and the scanning electron microscope. But first, let's just talk about a general overview of the electron microscope. The electron microscope is really great because it allows for higher magnification, over 100,000 times, and that allows us to visualize the smallest microbes, which are viruses. So up to 0.2 micrometers. And the electron microscope, as you would probably guess, uses a beam of electrons instead of light. This is how we get higher resolving power, because those electrons have a shorter wavelength than a regular light does. And so with the transmission electron microscope, the specimen has to be sliced super thin, and then those electrons are going to pass through that specimen. It's typically placed on some sort of mesh grid. It does have to be dried and fixed, so we can't view live specimens with this transmission electron microscope, and it can be magnified over 100,000 times, and because the specimens have to be so thin, sometimes it's hard to get a contrast between the specimen and the medium, whatever that they've placed it on, right? So sometimes they do use a stain, and those common stains are uranium, lead, and tungsten. However, these stains can distort the microbe when we look at it underneath the scope. So some downfalls, again, we can't view a live specimen, and it's very thin sliced, allows us to get a higher resolution, but we can't see a 3D view. And then because the specimens have to be dehydrated and fixed, then that distorts the microbe, and also we can't view ones that are live, like I said before. So the scanning microscope has a couple advantages for us then. One, the specimens don't have to be thinly sliced and prepared before viewing, and we can also get a 3D view. Another big difference between the two is with the transmission electron microscope, electrons are passing through the specimen, and with the scanning electron microscope, electrons move over the surface of the specimen. So that is what allows us to get that 3D view. And this image that you see here on the screen on the right is actually the COVID-19 virus, and they got that image by looking at it through a scanning electron microscope. So now we're going to talk about some different laboratory staining methods. And the first one we're going to talk about is called the simple stain. And a simple stain allows the microbiologist to see the shape of the bacteria and view some basic structures of the bacteria. And some common stains are methylene blue and carbofuchsin, safranin, and crystal violet. Methylene blue is also a dye that we use in surgery to identify structures. Sometimes fistulas or something like that we'll use methylene blue for. And then with the simple stain, they can also use something called a mordant, which increases the affinity of the stain, so it's more likely to stick to the organism. And so then we have differential stains. And differential stains are Gram stains. And Hans Christian Gram was the one to develop the Gram stain method in 1884. And with the Gram stain, a dye called crystal violet is used first, and then safranin is used. And the crystal violet is a really dark purpley blue, and the safranin is more of a reddish pink. And so if the bacteria have a really thick wall, then the crystal violet is going to stick to those, and they're going to appear like a dark purpley blue, and those are our positive microorganisms, are positive bacteria. And then if the bacteria has a really thin wall, then the crystal violet isn't really going to stick, but the safranin is going to stick. And so we're going to get this pinkish red kind of color that we refer to as Gram negative. Now there are some that have a tendency to change their mind, and those are called Gram variable, and one example is the bacteria that causes tuberculosis. And so then lastly, we have the acid-fast stain, and the acid-fast stain is used for classifying bacteria that are in the genus Mycobacterium, and that uses a bright red dye called carbofuchsin, and a counter stain called methylene blue. And acid-fast mycobacteria are going to stain red against a blue background typically. And so in this image here on the left, we have that Gram stain, and the dark purpley blue ones almost looks like some sort of maybe staphylococcus, some round little bacteria, some cocci bacteria, and then we have some rod-shaped or bacillus Gram negative bacteria. So here you can already see that this is helping us to classify and identify different types of bacteria. And then we have the acid-fast staining that you can see on the right side. To finish up our discussion about stains, we're going to talk about some special stains. And special stains are typically used for three reasons. One, for the negative staining for capsules, for spore staining, and for flagella staining. So for capsule staining, it's really difficult to stain these because the capsules do have a tendency to dissolve in water. So India ink and saffron is used, and it doesn't stick to the capsule, but it sticks to the background, stains the background. So then you have what's called a negative staining technique, where the stain is in the background, and then the colorless bacteria are highlighted because of that. And so the second one is spore staining. Remember, a spore is a way that a bacteria protects itself when there's like harsh environmental conditions, so it can kind of hunker down and weather the storm, and then it can return to its vegetative state after the environment is more suitable for it. And so when we do spore staining, again, it's really difficult to get the stain, the spore, to take up the stain. So all the other staining processes that we've talked about really won't work. So here they use something called malachite green, and they fix the spore, the bacteria to the slide, and then they go ahead and use that malachite green to do the staining. And then lastly, flagella staining. Typically, they use that reddish dye, that carbonyl fusion, to thicken the flagella, and they also use a mordant to help it stick a little bit better so that they can see those flagella. So here's a look at what those staining techniques look like. The first one is the capsules, so you can see how there's like this contrasting red background with a very dark core, and then that white surrounding each of those is the capsule, and the dark core is the bacteria. So it looks pretty awesome. And then in the center here we see the spores, and you can see ones that are in pink are in their vegetative states, and then the ones that are blue are the ones that have formed the spores, and they're in their dormant state. And then lastly, you can see these little creatures here with all of their flagella. So now we're going to talk about culture media. So when we're in the lab and we want to grow some microbes, well, we have to have somewhere to grow them. And culture media is where we do that. And so there's some things that we have to take into consideration with the culture medium. And we'll talk about those in a second. But first, I just want to let you know that the microbes that grow on the culture medium is what we refer to as the culture. Okay, so culture medium, it needs to be a happy place for the microbes to grow. It has to be an environment that is inhabitable to the bacteria. So if the bacteria like a certain type of nutrients like sugar, then it needs to have that. If they prefer a certain pH, or an environment with oxygen or an environment without oxygen, all of those things matter as well. They also have to be able to withstand the process of incubation, because that is a warming process, and it has to be able to hold up under those conditions. So interestingly, Robert Koch and his associate Walter Hess were credited with the discovery of agar, but it was really Hess's wife that gave them this great idea for something besides these liquid broths that they had been using. And so Hess's wife told him that her grandmother used to make jam, and she needed a way for it to stay solid in the hot tropical environment where she lived, and she used something called agar. And agar comes from red marine algae, and it's just a solidifying agent, and it's used in foods that we eat. It's just a polysaccharide, and it serves as the perfect solidifying agent for the nutrient broth that we are going to use to grow microorganisms. And so there's two ways that you might see it. One is in a petri dish, which is a round little dish like you see in the bottom image, and then the top image is called a slant. And that's a test tube, and they just kind of tilt it to the side so it gives this larger surface for the bacteria to grow on. Sometimes they do use test tubes where they just leave them straight up and down and the medium solidifies in there, and that's called a deep. So there's three different types of media that we're going to talk about, and they are enriched, selective, and differential. So let's go over some specific types of culture media, like I mentioned earlier. So we're going to talk about enriched media, selective media, and differential media. So this is the bacteria's food, right? It's some sort of nutrient broth, and we've added solidifier to it. Sometimes there are other components that are going to be added to it to either enrich the media so that these organisms that typically only grow in animals, or they won't grow outside of living cells, which we call fastidious cells. The media is going to be enriched with some delicious nutrients that are going to encourage those bacteria to grow there. And so a couple examples is blood agar and chocolate agar. And blood agar is a combination of agar and sheep erythrocytes, yum, and it is used to cultivate certain microorganisms like Staphylococcus, Epidermidis, Diplococcus pneumoniae, and Clostridium perfringens. And then the chocolate agar is mixed with nutrient agar and powdered hemoglobin, and this gives it kind of a chocolatey brown color. And it's used to grow the bacterias or viruses like Neisseria gonorrhea, Neisseria meningitis, and Haemophilus influenza. And so then we have selective media, and selective media, just like the name suggests, is going to select for a certain type of bacteria to grow there, and it's going to inhibit other types from growing there. Okay, so one example is the McConkey agar. It is selective and it's also differential, which means that it can help us distinguish between different types of bacteria. And when gram-negative bacteria are grown on the McConkey agar, they actually produce fermentation, and that makes these pink colonies of bacteria that make them distinguishable because of their lactose fermentation. And so then that helps to differentiate between lactose fermenting bacteria and non-lactose fermenting bacteria. And then another bacteria that grows well on selective media is Salmonella. So there are some other types of selective media that the book mentions, ones that select for gram-positive bacteria, ones that select for salt-tolerant bacteria or gram-negative bacteria. Also those that go through sugar fermentation selects for those as well. And then differential media. So differential media, again, helps us to distinguish between specific colonies of bacteria that are growing in the same dish. And so blood agar is often used to identify bacteria that destroys erythrocytes. And if the bacteria is going to destroy those erythrocytes, it's going to leave a red ring around that area. It's going to lyse those erythrocytes. And then lastly, here for differential media, the mannitol sugar agar, that MSA, only allows for salt-tolerant bacteria to grow there. And it usually fermentation occurs, and then this creates a yellowish colored colonies of bacteria. Okay, so two more types of media, reducing media, and then we're going to talk about rapid ID testing, which kind of uses a combination of media. So reducing media is used to prevent destruction of anaerobic bacteria. So anaerobic bacteria, they want to live in an environment where there isn't any oxygen. And so they will combine the agar with sodium thioglycinate, and that is going to combine with the oxygen and eliminate it. If they do have to grow the culture in a petri dish, then they have these special jars that they can use. And then, those scientists or microbiologists that work with anaerobes on a daily basis, they can use something called an anaerobic chamber, and this is a chamber where there is no oxygen, and they can insert their hands into these gloves and go ahead and work with and manipulate the cultures as they need to. And then lastly, rapid identification testing. These are basically kits that allow individuals, physicians, whoever, to test something on site and get pretty immediate results. The first type of rapid identification testing came about due to HIV/AIDS epidemic. And so now there's other ones that are used to rapidly test for strep infections, and there are ones that have multiple types of culture in one device. That's called the enterotube. And then there are other ones that are used for detecting anaerobic gram-negative bacilli, and of course they have some rapid testing machines for COVID now as well. All right, so here we're looking at some images of different types of media. So what about this first one? Any ideas? What does that look like? If you guess chocolate agar, then you would be right. It does look like a Hershey's chocolate bar right there. What about this next one here? Enterotubes, right? And what about the one on the end? What does that look like? If you said blood agar, then you are absolutely right. What about this contraption down here in the bottom? Looks like it has a place where we can put our hands. That is the anaerobic chamber. And then this bottom middle one is representing that McConkey agar. Remember, this is due to lactose fermentation, causes these bacteria to appear different colors. And then this last one here is that mannitol salt agar, or they abbreviate it as MSA, remember, and that's different types of bacteria will grow there that have an affinity for salt. So how do we make an anti-serum? Well, from anatomy and physiology, we already know that when a microorganism invades our bodies start producing antibodies in response to the antigens that the microorganism is going to secrete, right? So a similar process is used in the lab where solutions of antibodies are commercially produced, and then they're used to identify microorganisms that we don't know what they are or not. They're unknown, right? So there's two popular tests. One is the ELIAS, or the enzyme-linked immunosorbent assays, and then the other one is the slide agglutination test. So ELIAS is extensively used in the laboratory, and what they do is they take these little plates, and you can see one in the top image here on this slide, and they're going to place the antibodies in the depressions of this little plate in each of the wells, and then they're going to put the unknown bacteria, so different antibodies, and then the unknown bacteria, and if a reaction occurs, then they know they have a winner, right? And then the second way is by slide agglutination, and that image is down below. Remember, if bacteria agglutinates, it means it clumps together. So they're going to take some microscopic slides, and they're going to place the bacterium in a drop of saline, and then they're going to add different types of antisera until they get an agglutination reaction. And when they do, then they're going to know that the bacteria have an affinity for that antiserum. And so that's going to help them know what type of bacteria it is. Okay, so we're going to talk about some different types of laboratory analyses, and the first one is the catalase test. And the catalase test catalase is an enzyme, right? It causes some sort of reaction to occur. And so it's an enzyme that's produced by a bacteria, and it rapidly breaks down with hydrogen peroxide. So a culture is going to be obtained, and they're going to put it on a slide, and they're going to put some hydrogen peroxide on there. And then if it bubbles, right, that's what effervescence is, then it's a positive catalase test. If there's no reaction, then it's negative. So an example of a positive catalase test would be with Neisseria gonorrhea and Staph aureus, and then negative strep enterococcus. And secondly, let's talk about the coagulase test. The coagulase test is another enzyme that is produced by some bacterial species that coagulates blood plasma. Okay, so it's typically used to identify gram-positive, coagulase-positive Staph aureus, and this bacteria is extremely resistant to the inflammatory process of phagocytosis. So they're going to take this bacteria and they're going to put some on a slide. They're going to inoculate the slide with a culture and the prepared plasma, and they are going to wait and see if coagulation occurs. If it does, then the test is positive. Another laboratory analysis is amino acid sequencing, and this is used in determining how closely microbes are related. So the more similar the proteins in a microorganism, the more closely related they are. Phage typing, flow cytometry, and nucleic acid hybridization are all ways of helping us determine how closely related one microorganism is to another. And so the first one on the slide here is phage typing. Phage referring to bacteriophage. Bacteriophages are these little bacterial viruses. So they're viruses that invade bacteria, and they Shanghai their machinery to do their dirty work and eventually cause lysis of the bacteria while the viruses go skipping about. So what they do here is they're going to take a petri dish and they're going to cover it with a certain type of bacteria. And for this phage typing, some common ones would be Staph aureus, Vibrio cholera, Salmonella typhi, and Pseudomonas. And so they're going to cover the Petri dish with one of those types of bacteria, and then they're going to put some different drops of solution with the different types of phages in the solution around the Petri dish, right? And then they're going to wait and see if there's any clear spaces that develop as the as time goes on. And these clear spaces they refer to as plaques, where no bacteria has grown, and if that's, or where it has been destroyed. And if that's the case, then that means the bacteria is sensitive to that specific phage. So that way they can help to determine if a patient has a surgical site infection, they can take a sample of that bacteria, and they can also take a sample of one of the surgical team's bacteria, and they can identify if the two are the same. And a common one I mentioned earlier today was Staph aureus, and Staph aureus can become a resistant form, and it colonizes in the nose, which is one reason why it's important that we cover our noses with our masks. I see some people wearing them with their nose sticking out, and so they can test inside the nose of the surgical tech or the nurse or the surgeon or whoever, and they can test the bacteria in the wound and see if the two are the same. So the second one here, flow cytometry. For this, we don't need a culture. It uses a special laser sometimes and a computer to actually allows us to see the different shape of the bacteria and those kinds of things. Also can show the density and the morphology. And then nucleic acid hybridization is has to do with the way the strands of DNA are held together. And they're held together by these really weak hydrogen bonds. Remember, hydrogens really don't care if they stick to something or not. They're not really electromagnetic. And so they will use this hybridization procedure, and it's going to heat the strands of DNA and cause those strands to unwind. And then they're going to see if they have two different microorganisms, if one strand is going to, how well it will bind with the other strand. And that will tell them how closely related they are. They also have another process that involves RNA. Remember, mRNA is the copy of the DNA, and so they can also make a copy of the DNA and then take that RNA strand and see if it will bind to the DNA of the other organism, and if it does, or how well it does, can give them an indication of whether or not the two bacteria are related. All right, everyone, we are coming into the home stretch here, continuing to talk about some specialized laboratory analyses. Next up to bat is the nucleic acid base composition. And again, this is helping us compare two different types of microorganisms. Maybe there's been a patient that had a surgical site infection, and so we've taken some of that cultured that out, and we've cultured the surg tech, and we're going to see if those two organisms are the same. Might give us an idea of where this microorganism came from. And so here we are looking at the percentage of nucleic acids that are the same in each organism. So if there's more of a 10% difference in their guanine and cytosine is what they're specifically looking at, those bases. If there's a 10% or more in their percentage of pairs, then they're not going to be related. So for example, if one bacterium's DNA has 30% G's and C's and the other has 65%, they're probably not related. Right, so something else that they can do is they will do a restriction enzyme test, and they will take DNA from the two microorganisms and they'll treat it with the same restriction enzyme, and they're going to look for patterns as they develop, right, as the colony grows, and they refer to this as a DNA fingerprint. And the more closely related the two organisms' DNA fingerprints are, the more likely they are to be similar. And then the book talks about an example of at one hospital in the Midwest, they were having this high occurrence of patients that underwent coronary artery bypass graft. Cabbage were developing a post-op infection caused by a specific bacteria, and so they were able to use this DNA fingerprinting to identify that the source was a surgical nurse. And then there's kind of an extension of that is called the polymerase chain reaction test, which can occur in real time so that we're not waiting around to get the results. And this is in critical cases of drug-resistant strains. This is a good tool for us to use because it allows us to be able to treat more quickly and effectively. The hanging drop technique is used to assess bacterial motility. So the slides that we use for a motility test have like a little depression in them, and then that gets filled with fluid and it gets inoculated, and then you can view the bacteria. This was first used by Robert Koch in the 1800s. And then lastly, numerical taxonomy. So this is going to compare multiple factors, multiple characteristics. So it's going to look at shape, it's going to look at the organism's biochemical characteristics like its DNA and its amino acid sequences, G and C percentages, etc., etc. And if we get a match, a similarity score, you know, like some kind of bacterial dating website, if the match is 90% or more, then that typically indicates that it is the same species. All right, lastly, here's some images for us to take a look at. We're going to start on the top left, and you can see these little fuzzy bubbles, and this is the catalase test. Remember, it uses hydrogen peroxide, and if there is an effervescence, that means it's positive. If not, negativo. Okay, moving on, moving down, we have the coagulase test. Remember, that is going to see if bacteria coagulate or not. And if they do cause coagulation, that is a positive test. And Staph aureus is an example that gives us a positive coagulase test. Over here, top right, sorry about this, I used to use this in real-time class for like labeling and matching, so we're kind of jumping around here, but top right, amino acid sequencing. Remember, this is going to help us identify the evolutionary course of two organisms. All right, can help us determine how closely related they are. Let's see where we're going next. Top middle is the phage typing. Remember, phage typing meaning bacteriophage. These little viruses that like to invade bacteria and wreak havoc. So you can see these little plaques, these little dots that have developed on the slide. They're called plaques. That is going to tell us that that bacteria is sensitive to that type of bacteriophage. Bottom left, we have the flow cytometry, and the flow cytometry again is going to allow us to detect the difference in electrical conductivity between bacterial cells, and the bacteria is going to be traveling in a fluid, and a computer is going to be used in this case to analyze, the shape of the bacteria, how many there are, its density, yada yada. And then moving on, we have bottom middle. That's representative of the nucleic acid hybridization. So again, we have this top little image. This is our DNA strand held together by those hydrogen bonds. We heat it up a little bit. The two strands come apart. We take one of those strands. We take a strand from a different organism, and we see if they hook up. And if they do, then that gives us the indication that those two organisms are closely related to one another. Very center here, nucleic acid base composition. Again, we're looking at G's and C's, and we're going to see what the percentage of is in one organism versus another. And if there's less than 10% of a difference, that's going to give us a good idea that those two organisms are very closely related, if not the same. Bottom right, we have the hanging drop technique. You can see this slide has the little indent. We put the goods in there, and then we can go ahead and look at its motility underneath the microscope. Second to last here, we have the numerical taxonomy. And the numerical taxonomy, again, is going to look at morphology, biochemical characteristics, amino acid sequences, and percentage of G and C pairs. It's going to look at a bunch of different characteristics, again, to see if those two organisms are closely related or not. All right, and I think that concludes that discussion and that review. All right, so here we are at the end. I hope that this was helpful. We're talking about microbiology lab today. So thank you for listening, and I will be talking to you in Chapter 4.